Hey students, my name is Dr. Dave, Director of the Spark Science Program, and I'd like to thank you for watching this video today. Obviously, like many of you, I have been spending a lot of time in my home during this global pandemic, and I just want to say that I hope everyone is staying safe and healthy. I know these past few months have been likely very emotional. Whether you feel aggravated, sad, or another emotion, I'd like to remind everyone that it is critically important that we all stick to the recommended health guidelines to get us through this pandemic. One note before moving on to today's topic is my disclaimer. We are only offering a basic understanding of these topics. There are pages upon pages of textbooks that have been written on viruses and the immune system. And no matter how fast I read today, I'll never be able to cover every detail in this short video. Thus, this video is not comprehensive and is simply meant to serve as an overview to these topics. But be prepared, parts of this video are going to move kind of quickly. Over the past couple months, there is no doubt that you've heard and talked a lot about the coronavirus. Maybe you've even heard words such as COVID-19, SARS-CoV-2, or severe acute respiratory syndrome coronavirus 2. So what's the difference? Simply put, the term coronavirus is a general name given to a large family of viruses that can cause illness ranging from the common cold to respiratory problems in both humans and animals. They get their name from the crown-like spikes or coronas that appear on the viruses when looked at under a microscope. The term SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2 and is the actual name of this specific coronavirus causing the current pandemic. COVID-19, on the other hand, is the actual name of the disease caused by SARS-CoV-2. COVID-19 stands for Coronavirus Disease 2019, the year it was first detected. While practicing social distancing, mask wearing, and hand washing are critically important for preventing the spread of COVID-19, at this point, unfortunately, if we want to get the world back on track, we need to create a vaccine. But why? How will a vaccine help us get through this pandemic? Will a vaccine actually allow us to live without those silly masks? Well, it seems that I might be jumping the gun a bit, because before we begin learning about vaccines, we need a little more background information on two other topics, the coronavirus itself and the immune system. All right, let's first talk about this pesky virus we now know as Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2 for short. Here is a basic model of the coronavirus. Remember those coronas or spikes we mentioned earlier? Now I'd like to spend a few moments briefly looking at the virus in a bit more detail, including the parts that will become important when we talk about vaccine development later in this video. Surrounding the virus is the envelope. Its job is to keep the stuff inside separate from the outside world. The spike protein is anchored into this membrane and is directly responsible for virus entry into the host cell, and thus is a critical piece during viral infection. The hemagglutinin esterase dimer is the spike protein's partner in crime and aids in attaching to and invading host cells. Inside, we find the RNA, or the genomic material, of the virus. While humans store their genetic information in the form of DNA, the coronavirus contains another nucleic acid called RNA. Once inside a host cell, this viral RNA contains all the instructions required to make new virus particles. The M protein wraps around the RNA strand and helps to keep it safe. Now that you know a little bit more about the coronavirus, I'd like to spend some time talking about our immune system. You know, that not-so-simple network in our body that helps to protect us from being bombarded by germs and other pathogens? Wait, what's that word I said? Pathogen? The word pathogen can be used to describe any type of foreign invader that attempts to get into our body, such as a virus, bacteria, fungi, or worms. For the rest of this video, all pathogens will be represented by this little character here. Another term that you need to be familiar with is the word antigen. An antigen is any part of a foreign object that causes the body to induce an immune response. These could include different parts of a pathogen or various molecules on its surface. Oftentimes, a protein will act as the antigen of a foreign invader. In the case of SARS-CoV-2, the spike protein is often recognized as the antigen. For the immune system to defend our bodies against the onslaught of pathogens we encounter every day, it takes many lines of defense. For example, our bodies have non-specific ways from preventing infection, like our skin, mucous membranes, and hair. Another non-specific way our body fights infection is to induce an inflammatory response to help fight off the invading pathogens. Unfortunately, however, skin and inflammation are not enough to protect us from all invaders. The body also has very specific ways to fight off infections. This process is called the adaptive immune response. There are many organs and cell types that play a role in adaptive immunity, but for the purpose of this video, I'd like to introduce you to just a few. For starters, there are the antigen-presenting cells. The main function of the APCs is to process the antigenic material of a pathogen and present it on the cell's surface to other cells of our immune system called T cells. During the cell-mediated response, helper T cells are activated to release certain chemicals which then activate another cell type called cytotoxic T cells. These cytotoxic T cells spread throughout the body and hunt down not only foreign invaders presenting this specific antigen, but they also destroy other host cells that might be infected by the pathogen. The other type of adaptive immunity involves the humoral response. This process also involves helper T cells, which interact with naive B cells and stimulate plasma B cells. Plasma B cells produce what we know of as antibodies. Antibodies recognize and bind to specific antigens and flag either the antigen containing foreign or host cells for destruction. In both processes, another class of cells that are produced are known as memory B and T cells. As the name predicts, these specific cells keep a record of the antigens to which we've been exposed. Once the body recognizes a previous antigen, memory B cells can quickly activate plasma B cells to create antibodies again. Likewise, memory T cells can quickly activate cytotoxic T cells to seek out infected cells. So how does all this relate to vaccines? Well, the simple answer is that no matter which vaccine we're talking about, the goal is to artificially cause the body to induce the adaptive immune response, thereby creating these memory B and T cells we just mentioned. In the case of COVID-19, the purpose is to trick the immune system into activating an adaptive immune response to fight off the SARS-CoV-2 virus in the future. Just like practicing for your favorite activity over and over and over again until you're ready for prime time, vaccinations allow your body to practice for the real thing. In other words, a vaccine is meant to invoke a low-grade immune response, which ultimately creates memory T and B cells against the coronavirus that causes COVID-19, so they can attack the real virus should the body be exposed again in the future. Now, at this point, you might be wondering how scientists create a vaccine or what types of vaccines have been used in the past. In fact, scientists have taken many different approaches to creating vaccines. One kind is called the subunit vaccine. For this type of vaccine, a piece of the virus that is important for immunity, like the spike protein of SARS-CoV-2, is used to make the vaccine. The body can then generate an immune response to the spike protein so it can more quickly and efficiently fight off the virus down the road. Another type of vaccine uses the entire virus to create an immune response. But in this case, the virus is inactivated or killed with a chemical during the process of vaccine development. In this way, the body is able to recognize the antigens, but the virus does not cause a full-blown reaction. Similar to the inactivated virus, scientists can also use weakened live viruses to create a vaccine. Non-replicating viral vaccines is another technique. But this technique can be slightly confusing because it actually uses two different viruses. The first virus is a host virus that cannot infect humans any longer because it lacks the ability to make all of the proteins it needs to reproduce itself. However, it is able to make some proteins, including in this case, the SARS-CoV-2 spike protein. When the spike protein is made by the non-replicating virus, The body, or host, kicks off an immune response and prepares it for the real SARS-CoV-2 in the future. A similar approach is utilized when creating an DNA or RNA vaccine. In essence, scientists are injecting only the genetic material required to create a piece of the SARS-CoV-2 virus. Similar to the previous example, the DNA or RNA would simply encode for a piece of the virus like the spike protein. Our cells would take up the DNA, RNA, and using their own host machinery, create the spike protein, which in turn would be recognized as foreign by our bodies, thereby invoking an adaptive immune response. Many of you probably have tons more questions about COVID-19 vaccines. For example, you might be asking, well, which type of COVID-19 vaccine is most likely to work? Actually, there are over 170 vaccines currently in development. And in reality, it's likely that more than one of these approaches will work. However, until large clinical trials are completed to test for effectiveness as well as safety, we won't know which ones will work and are safe. In fact, these various approaches to vaccine development have different strengths and weaknesses. For example, the RNA or DNA vaccines can often be much faster to produce, but neither has been used to successfully make a human vaccine. On the other hand, subunit vaccines and live weakened viral vaccines have been used safely and effectively for many years. Unfortunately, however, they often take longer to produce. Many of you might also be wondering why it takes so long to create a vaccine. Well, first and foremost, scientists and doctors need to make sure the vaccines are safe for patients. Furthermore, biomedical research is tough, and there are a lot of unknowns throughout the process of vaccine development. For example, each type of vaccine could cause the immune system to respond differently in different individuals. Understanding the immune responses in various populations of people will be important for determining distinct advantages, like whether additional booster doses will be needed, how long vaccine recipients can be protected, and if one type offers benefits over another. Finally, everyone on Earth is most likely going to need a COVID vaccine. So as you might be able to guess, it is going to take a while to ramp up production. Of course, creating an effective and safe vaccine is the ultimate goal for getting our world back to normal. While the SARS-CoV-2 virus might not be going away anytime soon, with the help of a vaccine, we can significantly reduce the amount of illness and death that occurs. In the meantime, we can still benefit from social distancing, washing our hands, and wearing a mask. As scientists and healthcare professionals continue to work toward a solution, I hope everyone remains healthy and safe. Thanks again for watching, and have a great day.